The overall goal of this procedure is to assess individual CFTR function and efficacy of CFTR modulating treatments using a Forskolin-induced swelling or FIS assay in intestinal organoids generated from cystic fibrosis patients.
This method addresses key questions in the cystic fibrosis field. Most notably, it helps the identification of patients that can benefit long-term from existing or experimental drugs. The main advantage of this technique is that easily accessible tissue can be used to generate organoids from any CF patient, independent of age.
The implications of this technique extend toward the therapy of cystic fibrosis and addresses an urgent current need for testing drug efficacy in a cost-effective and individual manner.
Demonstrating the procedure will be Marvin and Statia from Hubrecht Organoid Technology and Annelotte Vonk from Jefferies Beekman Group.
For obtaining optimal Forskolin-induced swelling assay data, it is essential to work with a culture containing big organoids with multiple buddings. After identifying such a culture, label one 15 ml conical tube with the sample name and one tube as washed. Next, use a P1000 pipette to carefully aspirate the medium from the organoid culture wells without disturbing the basement membrane matrix drops and add 1 ml of basal medium to each well. Now use the pipette to break up the basement membrane matrix drops in each well and transfer the resulting organoid suspensions to the 15 ml tube with the sample name. Rinse each well with another milliliter of basal medium and pool the washes in the sample tube.